two DNA strands intertwine to form a double helix. Each strand has a backbone composed of phosphates and sugars to which the bases are attached. The bases form the core of the double helix, while the sugar-phosphate backbones are on the outside. The two grooves between the backbones are called the major and minor groove, based on their sizes. Most protein DNA contacts are made in the major groove because the minor groove is too narrow. The DNA backbone is assembled from repeating deoxyribose sugar units that are linked through phosphate groups. Each phosphate carries a negative charge, making the entire DNA backbone highly charged and polar. A cyclic base is attached to each sugar. The bases are planar and extend out perpendicular to the path of the backbone. Pyrimidine bases are composed of one ring and purine bases of two rings. Adjacent bases are aligned so that their planar rings stack on top of one another. Base stacking contributes significantly to the stability of the double helix. In a double helix, each base on one strand is paired to a base on the other strand that lies in the same plane. In these base pairing interactions, guanine always pairs with cytosine and thymine with adenine. A GC pair is stabilized by 3 hydrogen bonds formed between amino and carbonyl groups that project from the bases. In contrast, an AT pair is stabilized by 2 hydrogen bonds. The specificity of base pairing, that is, C always pairing with G and A always pairing with T, ensures that the two strands are complementary. This is important for DNA replication and transcription.
But where were the genes? What genes are made of remained a mystery, but some scientists thought they knew where they were. Chromosomes, curious filaments in the nucleus of each cell, behaved a bit like genes. You get one from each parent and pass one on to each offspring. Chromosomes contain protein, as well as a rather boring molecule called deoxyribonucleic acid, DNA for short. Most people were convinced that if chromosomes held the genes, then protein was the place to look because nothing else was complicated enough. Fred Sanger is the only person ever to have received 2 Nobel Prizes in the same subject. He remembers just how unpromising DNA seemed.
When I started working in the 1940s, DNA was thought to be just something that within the nucleus and probably a structural thing, but the composition of it was much too simple to imagine that it had much biological activity.
This is Tübingen in southern Germany, and its castle was the unlikely setting for a breakthrough in molecular biology. Friedrich Miescher was a young researcher stationed at the laboratory here. On the advice of his uncle, he studied the cell nucleus. His experimental material was white blood cells taken from the pus-soaked bandages of wounded soldiers at the local hospital. In the nucleus, he found protein, but he also isolated another substance, a long, sticky molecule that he noted was rich in phosphorus. He called it nuclein. We call it nucleic acid. He was the first to discover DNA. This would have been remarkable enough, but Miescher went on to suggest how it might be used in heredity. In a letter to his uncle, he wrote that molecules of DNA would be quite capable of carrying all the richness and all the variations of hereditary transmissions, just as all the words of all languages can be carried by just 24 to 30 letters of the alphabet. His prediction of the genetic code came in 1892, but Miescher did not live to see it accepted. The freezing laboratory contributed to his early death from pneumonia, but in any case, it would be another 60 years before science was ready to accept that DNA carried the genetic code. The breakthrough was in 1953, thanks to Cambridge scientists Francis Crick and James Watson in the most celebrated research of the 20th century. Using the X-ray pictures of DNA taken by Rosalind Franklin and Maurice Wilkins, they struggled to make the data about the spaces between the atoms fit into a structure that made sense. When they had finished, it did make sense, but in a more profound way than the structure of any molecule before or since.
The most important thing about that structure was not that it was a double helix, which is the icon of the 20th century we all are aware of, but the fact that it was a very long linear molecule and that these bases, the AGCT symbols, were distributed linearly along it. And they spotted and confirmed the notion that people have been fighting against in a way, that it was digital, because it seemed unreasonable that something sort of soft and squidgy and lifelike like us would somehow be coded for in this hard digital fashion. But it is so, and that's the breakthrough which now we're exploiting today to actually read out that code.
Although the double helix looks complicated, what it does is very simple. Crick and Watson had opened up a world not of chemistry but of information. Each strand of DNA carries a set of 4 bases, abbreviated to A, C, T and G. The bases can occur in any order along the length of the molecule, and this is how the information is carried, just as the letters of the alphabet make words. But the crucial thing is the way the bases on opposite strands are joined. Chemistry allows A only with T, C only with G.
And they saw also, and this was the famous sentence, "It has not escaped our attention that..." This provides a mechanism for replication, because they saw that if you pulled the 2 halves of the double helix apart, then used those same best pairing rules to reconstruct new strands, you would have 2 new double helices looking just like the old one. You had replicated DNA, and as Francis Crick said in the Eagle Pub, that was the secret of life.
Crick and Watson realized that each strand could act as a template for the other. If you split them apart into 2 single strands and then throw in a mixture of A, C, T and G, the bases can only join up in the right places: a T opposite every A, an A opposite every T, a C opposite every G, a G opposite every C. And you have accomplished one of the great tricks of nature. You've taken one copy of the code and made 2 identical, perfect copies that can pass on to the next generation. Through chemistry and physics, Crick and Watson had discovered how the instructions for life are passed on.
If you're going to build anything complicated, you need some sort of plan, a blueprint, a set of instructions. Now that's just as true of living cells as anything else, but they contain the instructions within themselves. They're inherited. They're copied and passed on from generation of cell to generation of cell. Now what we're going to do is look at what the instructions are and how they work. But before we do that, just stop for a moment and think what sorts of characteristics any set of inherited instructions is likely to have. I think there are probably 3. First of all, the instructions have to be stable. They have to last long enough to be copied and passed on to the next generation of cells. And there's the second point. They need to be capable of being copied, and copied accurately. And finally, and most obviously, as instructions, they need to contain information. Well, that's all very well, but it's really just sort of speculation, theory. What about the reality? The reality is a molecule called DNA. Now you might think that the structure of a molecule that contains the instructions for making a complete cell must itself be very complicated. But in fact, the basic structure of DNA is remarkably simple.
Here's a schematic model of DNA. Unwind the double helix, and it consists of two long strands that form a ladder-like structure. The strands of DNA are strings of chemically repeating units, which act as basic building blocks. Each unit contains a sugar, deoxyribose, a phosphate group, and a base. Together, these form a nucleotide. There are actually 4 types of base: adenine, thymine, cytosine, and guanine. Within a strand of DNA, the bases can come in any order, and just how important this sequence is will become clear later. What's more, in double-stranded DNA, the bases match up in a particular fashion. Adenine always pairs up with thymine, and guanine with cytosine. This precise base pairing means that the base sequence in one strand is complementary to the sequence in the other. The base pairs are held together by relatively weak hydrogen bonds. But when summed up over the whole DNA double helix, these hydrogen bonds impart great stability.
Let's have a look